Hi, my name is María Fernanda Cerini. I'm doing my master's degree at the University of São Paulo in Brazil, and I'm performing all of my experiments at the Brazilian Synchrotron Light Laboratory. So my main focus is on molecular biosignatures. So one of the main questions we try to answer is about the possibility of life somewhere else in the universe. So how do we look for that? We need to find some evidences of the existence of life, either past or present. So that's what we call biosignatures. So they can be of a lot of natures, but elementals, molecules, morphological structures, or even phenomena, but I'm focusing on the molecules. So what type of chemistry should we look for? Well, I'm sure by now, after many lectures this week, you're all convinced that it should be carbon-based, it should be an organic chemistry because of the chemical versatility of carbon and ubiquity in the universe. Also, we should have large molecules so we can get a faithful propagation of our living organisms. And of course, it's the only form of life we know yet. So it's based on carbon macromolecules. So we should build a database of biosignatures, of biomolecules that could be potential biosignatures to facilitate their detection in another planets. So this recent work here, presented a few good candidates for chemical biosignatures, and two of them were like lipid keratin-derived compounds and porphyrin for being resistant molecular fossils. And also, if we look at biological pigments, they are very abundant in all terrestrial life forms, and they all have colors, so it should be easy to detect with the spectroscope. So we know that in astrophysical environments, the star's radiation is key to the evolution of organic molecules, and also in excess it could be essential for the degradation. So it could destruct the biosignatures we're looking for, or transform them. So it's important to assess the photochemical role of the star's radiation on the formation, transformation, destruction of the biomolecules to give us insights about their stability of complex organic molecules in space and planetary environments. So how do I do that? Mostly I perform laboratory simulations at the synchrotron lab. And also we can use analog environments with high-altitude balloons and real space environments with CubeSats. So the samples based on those candidates, we chose beta-carotene and chlorophyll A, which are both biological pigments, and I mixed those with substrates. We have two phyllosilicates, which kaolinite and montmorillonite. They are both hydrated minerals. They are well-known catalysts for organic synthesis, and they were already implicated in the early solar system organic reactions. They could have aided the construction of macromolecular materials. So, and also they have been identified on Mars surface. Also, we have regolith simulants. They are mostly oxides, and they are volcanic ash altered at low temperatures. So I used simulant of the lunar regolith and the Martian regolith. And the spectroscopic techniques I used was UV reflectance. So here we have a chlorophyll A, we have 2 peaks, and for beta-carotene we also have 2 peaks, but usually I can detect only one of them. And also I use RAMAN spectroscopy using the laser with 785 nanometers. But with this laser, I can only see beta carotene, those 3 peaks. And chlorophyll, we have too much fluorescence, and we can see the peaks. So, to the simulations, this is the Brazilian Synchrotron Light Laboratory. Douglas already talked about a lot. And I used this beamline, TGM beamline, which gives off synchrotron radiation. I use the white beam, so it's the polychromatic. But this beamline is optimized to work in the VUV region. So this combined with the vacuum chamber, we can do space environment simulations. And these are my samples. Here we have beta carotene mixed with all the substrates I told you, and before and after the radiation, we have the spot here. And also for chlorophyll, the same, and here we can see the spot for the synchrotron beam. In these experiments, I used in situ the diffuse reflectance spectroscopy. Basically, I put my sample in 45 degrees with the white beam, and then I collect them with an optical fiber using a spectrometer, and I read their reflectance in real time. So after 1 hour of irradiation, this is one of the samples, the chlorophyll A with the lunar regolith. So we can see clearly a decrease in the reflectance of my spectrum. I use the lunar regolith as the reference. And how do I calculate this? We have a problem at the synchrotron because the current decreases with time. So we have this change in the baseline. So to correct that, I just do like that. I monitored 3 points of the band, and then I calculate it's like a delta in the intensity. So this delta, it's the thing I'm putting this in the y-axis, and then I plot with the fluence I'm giving off with the synchrotron light. This is another example, the beta carotene with the same regolith, and we have a decrease also in the reflectance, and also I had to try to degrade the substrates just to show that what I'm degrading is the samples, not the substrate. So for the lunar regolith, for example, we can see that they don't change after time with the fluence. So, okay, so after I did that with all the samples, I'm just presenting all the beta carotene, just the 45 nanometer peak, decreasing with time, with the fluence for 5 substrates. So we can see for the 2 peaks on the chlorophyll A as well, 437 nanometers, and also 667 nanometers. And this is for after a fluence of about 1.8 10 to the 6 joules per meter square. So the simplest way of looking into these experiments, these results, is simply taking these last points and see how much they degraded, and then I put this in one slide, in one graph. So we can clearly see that the pure form is degrading more than the others. And then we have different different things for different peaks. And usually we would hope that the regolith simulants would protect more the samples, but it's clearly not the case. But then we could look into better ways maybe of analyzing this. So I tried to perform exponential fittings. And it's not possible to do one exponential fitting for the whole graph, the whole curve. So what I tried to do was to break into 3 steps. So this is for the beta carotene, and this is for the chlorophyll A, for example. And then we call this maybe first stage of degradation and second stage of degradation and a third. And with the breaking points of fluence in which maybe they are degrading and degrading with different mechanisms, or we don't know. But then looking just only into this first stage, we could plot this rate of degradation against the breaking point, the fluence of this breaking point. So we can see, for example, for beta carotene, that the results are different than the one I've shown before, and it's more likely that it's more what we expected, because the pure sample is degrading much faster than the others, and the lunar and Martian regolith simulants are maybe protecting them more. And then we see to chlorophyll A 2 different peaks. Also, the lunar and Martian are protecting more, and then here we can't say much about that because of the errors, but it's one way of looking into this. And also, I did the RAMAN only for beta carotene samples. So I used a micro RAMAN, 785 nanometers, and an average of 30 points at the same sample, at that same spot. So here we see in black we have the spectrum before the irradiation. This is the average. And after the radiation in red, so we can see maybe some decrease. And here is only a diamond powder, which is robust, chemically inert, that I used only for reference, for internal reference. So here is the 3 peaks of the beta carotene. Well, this one is the stretching and a combination of bending and rocking. So also maybe we can see that the pure form is degrading more, but we see different aspects for each peaks. But the interesting thing is that for more irradiation, we still can see the peaks, and we can then play with the results comparing to real environments. For example, the TGM beamline gives off this fluence in this energy range. So the surface of Mars would have this irradiance, so it would take about 9 hours at the surface of Mars, and on the surface of Moon, and even less, 3.8 hours. So in conclusion, both UV reflectance and Raman are potential for the detection of biological pigments, but for higher fluences, we can see that the UV's band it's disappearing, so Raman is more sensitive. And for the degradation stability studies, we see that maybe Raman is not ideal because we have a lot of surface dependence, and maybe UV's reflectance is more sensitive, but to look into what is really happening, we could do a lot of other analysis. So just to quickly say that our results also can assess the development and optimization of techniques and procedures for space missions applications. We can expose these molecules to real space environments, and also for the actual detection of biosignatures on other surfaces. So it can use analog environments, such with high-altitude balloons, which they're thin plastic balloons used for scientific research, and it has low cost and high turnaround rate, and it goes up to the stratosphere, about 20 to 50 kilometers above sea level, where we have low pressure and temperature, dryness, and high radiation. So it's a good analog for the Martian surface. We work together with students from São Carlos who built this gondola, and we sent this one at the beginning of last year, where we sent microorganisms and the biomolecules. And also in December, we sent another one. Here we have a lot of microorganisms and biomolecules. So these experiments, they work for, they're also good for preparing for real space environments using, for example, CubeSats, which are also small, light, low cost, and low energy payloads, where we can do for space and planetary missions, where we can go up to the LEO, the low Earth orbit, or even in the deep space, where we can, where we have the full solar spectrum, vacuum, cosmic rays, temperature fluctuations, and no gravity. So we have a real astrophysical environment. So me and my group already had, last year, these two companies from England, together with ESA and UK agency, they led contacts for 6U CubeSats to orbit around the moon. So we are participating on this mission, and with a lot of other Brazilian companies. This is the Garatea mission, and the proposed orbit is beyond the protection of the Earth's geomagnetic field. So where we can have where we are not protected from the cosmic ray charged particles, mostly from solar wind. So we are doing the astrobiology experiments, where part of the experiments will assess the effects on microorganisms, and part in the molecular level, where I'm gonna contribute. And we're going to expose the biomolecules to deep space conditions, with and without the protection, and also assess the molecular responses measured by in-situ and real-time spectroscopy. So that's it. That's my references. Thank you.